Hello, my friend. My name is Joshua Mugabi. I welcome you to Biology I Chew. Today we are going to look at genetics in DNA part 2. So without wasting time, let's go to our quiz. The bases in DNA form a triplet cord. What is meant by a triplet cord? It's a sequence of 3 bases that code for one amino acid. To which structures in the cell does mRNA carry the code? Ribosome. Give the role of the enzyme RNA polymerase. Joins nucleotides together to make mRNA. Where in the cell would you expect to find most DNA? In the nucleus, in the chromosomes, and also in genes. DNA contains the instructions needed to make protein. These instructions are called the dash code. Gene or genetic. During translation, one end of a tRNA molecule attaches to an mRNA codon. What is attached to the other end of the tRNA molecule? An amino acid. What is meant by non-coding DNA? Does not code for a protein or for RNA. Give one structure difference between DNA and RNA. DNA contains thiamine or RNA contains uracil. In DNA profiling, DNA strands are cut into fragments. On what basis are these fragments then separated? Size or use of gel electrophoresis? True or false? The base uracil is found in DNA. It is false. Each mRNA codon specifies one of the three possible outcomes during protein synthesis. Name the three possible outcomes. Start. Another one, adding an amino acid. And the other one, stop. Explain the term replication in relation to DNA. Making a copy. In DNA profiling, what are used to cut DNA strands into fragments? Enzymes. The triplet code is transcribed into mRNA. What does this statement mean? Information, or what we call the code, is copied to RNA molecule. In which structures in the cell does translation occur? Ribosomes. What does the letter T stands for in tRNA? Transfer. Where in a cell are enzymes produced? Ribosomes. Explain the term transcription in relation to DNA. Complementary RNA production. What must happen to the newly formed protein before it can begin to work? It must fold into its functional shape. Give two applications or uses of DNA profiling. Can be used in crime or what we call forensic, in medicine, in paternity, archaeology, and in evolution. What is meant by genetic screening? Checking for the presence of a specific gene. True or false? DNA is a double helical shape. True. Where in a cell does transcription occurs? Nucleus. Name the small biomolecules that are joined together to make a protein. Amino acids. Hello, my friends. Thank you very much for attending to Biology I Chew. Today we have been talking about genetics, generally about DNA, part 2. Let us meet in other Biology I Chew, and that one will be genetics, part 3. I'm Joshua Mugabi. Bye-bye for now.